Welcome to our today's lesson where we'll be talking about the Krebs cycle, okay, which is also sometimes referred to as the TCA cycle, which simply denotes or stands for the citric acid cycle, okay. So now this process is referred to as a cycle because the intermediate that we're going to start with is actually the same intermediate that we're going to do what to end with. No wonder it is referred to as a what? As a cycle, okay. What we're going to start with, it is actually the same that we're going to do what to end with. No wonder it is a cycle. It is not a cycle because I've written it in a cycle form or in a cycle way. No, no, no. Okay. So the Krebs cycle, this process actually happens where in the mitochondria of a cell. So this diagram that we are seeing here, it is just representing a what? A mitochondrion of a cell, of course. Okay. So now this process starts by or with a acetyl group or acetyl CoA. So it is acetyl CoA that will enter the Krebs cycle and restart the what? The cycle. Okay. So now acetyl CoA is actually being derived from the pyruvate. These are the 3 carbon molecule. Okay. This we call it the what? The pyruvate. So now the pyruvate here, it is 3 carbon molecule. In my previous video, when we were looking at glycolysis, we saw that glucose was being broken down to form what? Pyruvate. So now it is this same pyruvate that again will further undergo different processes so that it gets to do what? To be converted to acetyl CoA. So that acetyl CoA can now enter the Krebs cycle. Okay. So I've said that this is the pyruvate. Okay, this is the pyruvate. As you have seen, the pyruvate is actually a 3 carbon molecule. So now, if the pyruvate is a 3 carbon molecule, what happened to pyruvate if what is being produced from pyruvate is having 2 carbon atoms? So this is a 2 carbon molecule. So what happened is there was actually a loss of what? A carbon atom there in the form of what? Carbon dioxide. So anytime we lose carbon dioxide or anytime we lose the carbon atom, we're going to be losing it in the form of what? Carbon dioxide. So here what was happening is decarboxylation reaction. Okay. So what we did is we decarboxylate the what? The pyruvate. Okay. So the pyruvate molecule here was decarboxylated. So here there was actually a removal of carbon dioxide like that. So this carbon dioxide was removed. The process is decarboxylation. So now, in the process, as you have seen, the pyruvate, it is not having the CoA, but here the molecule that is being produced or that is being made from pyruvate, it is having the CoA. So in short, actually, in the same process, we did what? We added the CoA. So here, let us also write that. So we added CoA. So CoA was introduced. So that coenzyme A, or maybe I can just use a different color. So this coenzyme A was introduced in the what? In the reaction of the pyruvate and the acetyl CoA, so that we produce the what? The acetyl CoA. But again, the enzyme that is used here, we call it the pyruvate dehydrogenase complex. Okay, so we call it that because it is composed of 3 different enzymes. Okay, so now this same pyruvate dehydrogenase complex, what it does is, since it is a dehydrogenase, it means it is also going to produce what? NADH. Okay, so again we are producing NADH from NAD plus. So NAD plus is yielding NADH. Okay, like that. So this is what is happening here. So pyruvate is being converted to acetyl CoA by the help of different coenzymes or different cofactors. Okay, like for us to have to remove carbon dioxide, there is actually the vitamin derivative that is supposed to be used here, and for us to introduce the same CoA that you are seeing there is actually a cofactor that we need to use, and for us to have the NAD plus in place here is actually the what? The cofactor that we need. Okay, so now let us also talk about the what? The same cofactor, so that we understand what is happening here before we dive right into the what? The Krebs cycle. Okay, so what is happening here is this: for the carbon dioxide to be removed, okay, for the carbon dioxide, this carbon dioxide to be removed, there is actually the vitamin derivative, vitamin B derivative here, and the vitamin B derivative that we're talking about here is vitamin B1. Okay, the vitamin B1. That derivative of vitamin B1, we call it the thiamine pyrophosphate. Okay, thiamine pyrophosphate. So it is pyrophosphate. Pyrophosphate. Now it is also sometimes referred to as what? TPP, because it is thiamine pyrophosphate. Okay. So now it is that one. It is a derivative of vitamin B1. What of NAD plus? For us to have NAD plus in place here, we need to have also another vitamin B derivative, which is now coming from vitamin B3 derivative. So that one, it is vitamin B3. Okay. So this vitamin B3, we simply term it as a what? As a niacin, because this NAD plus is of course from what? Is derived from vitamin B3, which is niacin. Okay, niacin like that. So this niacin is of course the vitamin B3 derivative. Okay, so these are actually the cofactors that help this process to be driven. What of coenzyme A? So for us to have this coenzyme A, there was actually an acid, which is lipoic acid. Okay, that lipoic acid will also have the other derivative of vitamin B, which we simply call the pantothenic acid. Okay, pantothenic or pentothenic, pantothenic acid. Okay, so this is actually the vitamin B5 derivative. Okay, so pantothenic acid was helping us, or was helping this process to do what? To bring this CoA to existence, to that process, so that it gets to what? To be converted to acetyl CoA. Okay, so pantothenic acid was just bringing the CoA to the what? To acetyl group. Okay, so that now it gets to what? To bind that CoA to the what? To that acetyl group, so that we have the acetyl CoA. Okay, so this molecule that we have produced here now, we are going to call it the acetyl CoA. So this is the acetyl CoA. CoA like that. So now, since we have the acetyl CoA, the very important intermediate in the what? In the Krebs cycle, because it is this same one that will do what? That will enter now the Krebs cycle. Okay, so now let us talk about the different processes that will be happening here, different enzymes that are involved, and of course, different intermediates that are involved, and what we're going to be producing in the process. Okay, so this acetyl CoA, the moment it enters the Krebs cycle, it is going to be converted to a intermediate that we call the citrate. Okay, so this intermediate, it is having 6 carbon atoms, 1 2 3 4 5 6. So this is actually the citrate. Okay, that is citrate. So now this citrate was converted from acetyl CoA by the help of which enzyme? Okay, so we are just producing, or we are making citrate. So the enzyme, we're going to call it what? Citrate synthase. Okay, citrate synthase. The same name, citrate, but you just put synthase. Okay, that is basically the enzyme that helped us to do what? To produce citrate here. And we know that citrate is actually a 6 carbon molecule. So now this same citrate synthase, after it has produced this molecule, which we are calling citrate, it is again going now to convert, like this citrate will be converted to another intermediate, which we call isocitrate. Okay, isocitrate, but of course by a different enzyme, not the citrate synthase, because here this is what? Isocitrate. So that enzyme that is converting citrate to isocitrate, we simply call it what? Aconitase. Okay, aconitase. So it is aconitase, this enzyme aconitase, that is able to produce isocitrate from citrate. Okay, so now the conversion, how it happens is this aconitase, it will add water, here, H2O. Again, it will remove water. Okay, it will remove water. In the process, it is just doing what? It is just shifting the hydroxide or hydroxyl group that are where in the citrate, so that it gets to be what? To be made to isocitrate. So the aconitase, it is just shifting, okay, as it is moving the water into the process, and again removing the same water from the process, it is shifting the hydroxyl groups of the citrate. Okay, so that citrate can now be converted smoothly to isocitrate. Okay, so aconitase, it is basically doing what? Converting citrates to isocitrate. So here is actually a hidden process that happens. what it does is the moment it adds this water, okay, the moment aconites adds the water here to the citrate, this citrate we're going to call it cis citrate. Now, the same cis citrate, this cis citrate, after it has removed water, aconites after removing water, this cis will be what will be removed and it will be converted to isocitrate. Okay. And again, isocitrate, it is just basically having the six carbon atoms, just as the what? The citrate molecule. So now the isocitrate again to be converted to another intermediate by again a different enzyme. So isocitrate will be converted to what we call alpha ketoglutarate. Okay, so this is a different intermediate. So this is alpha ketoglutarate. Okay, glutarate. Alpha ketoglutarate, like that. Okay, so this alpha ketoglutarate, it is having how many carbon atoms? Let us count. We have 1, 2, 3, 4, 5. So it is having 5 carbon atoms. So what happened in the process here? So we can tell that actually what happened is there was a removal or a loss of carbon dioxide, because we are having 5 carbon atoms, because this molecule that this guy is being produced from had 6 carbons, okay, 6 carbon atoms, but this one is having 5. So what happened is there was a loss of what? 1 carbon atom from there in the form of what? Carbon dioxide. So here we are producing carbon dioxide. So carbon dioxide is being released there, okay, so that we have 5 carbons to this the same alpha ketoglutarate. So now the enzyme that was used here, because we are removing the carbon dioxide and this enzyme was isocitrate, and rather the enzyme here was aconitate and the molecule was isocitrate. So the enzyme that will be used here, we're going to call it isocitrate dehydrogenase. Okay, isocitrate dehydrogenase. So the moment you hear the dehydrogenase enzyme, just know that we are also producing what? We are also producing the NADH from NADP or NAD plus. Okay, anytime you hear dehydrogenase, that enzyme is associated with the production of NADH. Okay, so in some process that happens in the body, you are going to find that it is not necessary that NAD will just be like involved with that, but also the NADP plus. Okay, but for this one, just concentrate with what? NAD plus, and of course, yielding the NADH. Okay, so here the carbon dioxide is being removed, and of course, it is by the help of which enzyme? The citrate dehydrogenase. Okay, dehydrogenase. So that is basically the enzyme. So now the isocitrate dehydrogenase, because it is a dehydrogenase, we know that we are producing what? We are producing the NADH from what? NAD plus. Okay, so here NAD plus was used to yield what? To yield NADH, like that. Okay. So again, we are going to move from alpha ketoglutarate, we're going to move to a different intermediate again. Okay, alpha ketoglutarate will be moved now to a different intermediate, which we call succinyl-CoA. Okay, so this molecule is having 1, 2, 3, 4. It is also having 4. Okay, this had 5, this is having 4. So this one, we are going to call it a what? Succinyl. So this is actually succinyl-CoA. So if it is a succinyl-CoA, meaning even the carbon atoms, this molecule, it is actually attached to a what? A CoA molecule or CoA group. So here we're having a CoA like that. Okay. So this is actually the intermediate, the next intermediate from alpha ketoglutarate, we are having succinyl-CoA. So now the enzyme that helped us to move from alpha ketoglutarate to succinyl-CoA, it is actually the alpha ketoglutarate dehydrogenase. Okay, actually you can even try to relate, because here we had isocitrate. The moment we were shifting from isocitrate to alpha ketoglutarate, the enzyme that we used was the isocitrate dehydrogenase. So in short, we were just dehydrogenating the what? The isocitrate. So here what we are dehydrogenating is the what? is the alpha ketoglutarate. No wonder we are using alpha ketoglutarate dehydrogenase. Okay, and the moment you hear the enzyme dehydrogenase, that's one that we are also producing the what? The NADH from NAD plus. Okay, so here the enzyme that was used, maybe I just use a different color here. The enzyme that was used was alpha ketoglutarate, okay, alpha ketoglutarate like that, then dehydrogenase, okay, dehydrogenase. So that is basically the enzyme that was used there. Or to do what? To remove the, even the CoA here will be what? Will be removed, of course, okay, in the next step. But here we're just producing the what? The succinyl CoA. And in the process we are producing what? We are producing NADH from NAD plus. And the moment you count these carbon atoms, there are 1, 2, 3, 4, and here we had 5. Just know that we also produce what? Carbon dioxide. So in the process, there was actually the production of what? Carbon dioxide. So even there, carbon dioxide was produced. Okay? So carbon dioxide was released. Okay, was released in the process so that we produce what? We produce succinyl CoA like that. Okay. So now here again, succinyl CoA will be now converted to another intermediate. Okay, to a different intermediate, and of course by different enzyme. So succinyl CoA will now be converted to succinate. Okay, just succinate. So this molecule here we call it succinate. Okay, so this is basically the succinate molecule. So that succinate molecule, if you count how many carbon atoms are there, 1, 2, 3, 4, 1, 2, 3, 4. So now you can now try to count from here, from this succinate to all the intermediates that are going to be producing here. The intermediates after succinate, they are all having what? Same carbon atoms. Okay, so it is now very easy to do what? To understand. Yeah. So here, this succinate was converted from succinyl CoA to succinate by which enzyme? Okay, so here the enzyme that was used is succinyl CoA synthetase. Okay, succinyl CoA synthetase. Succinyl or succinyl, okay, succinyl CoA synthetase. Okay, that is basically the enzyme that was used here. Synthetase. Now, here we are having a synthetase, and this synthetase, it is producing what? The succinate. Okay, succinate, succinyl CoA synthetase is producing the what? The succinate. Now let us go back here. So here we had an enzyme that we were calling synthase, and synthase was producing what? Was producing the same citrate. Okay, from acetyl CoA we had citrate synthase. So synthase is producing, and even the same synthetase is producing. The only difference is that synthetase is using what? Energy. Then synthase is not using energy. No wonder there's actually a difference in what? In names, but they are both doing what? Producing something. Okay, synthase is producing, and of course synthetase is producing, and synthase is doing what? Is producing. So this one is actually involving energy. Now here the form of energy that will be involved is GDP. Okay, so GDP will be converted to GTP. Okay, so here GDP will be produced, GTP will be released from GDP. Okay, so that is actually the energy that will be converted there. But sometimes we can also produce ATP just directly, or the same GDP can be converted to what? To ATP later. Okay, so that can also be converted to ATP later. Okay, ATP. So because we're using energy, no wonder we're calling it what? Synthetase. Okay, so now since here we've produced the succinate, this again intermediate are going to also undergo further processes, and if it undergoes further processes, we're going to produce this molecule here. So this molecule, we simply call it a what? This molecule is called the fumarate. Okay, that is basically the fumarate. So this fumarate, the enzyme that was used for us to convert succinate to fumarate, we were just using dehydrogenase. And I said that each time you hear the word dehydrogenase, just that we are producing what? We are producing NADH from NAD+. But here it's a different case. Okay, we're not producing NADH, but we're producing FADH. Okay, so this is actually the so the enzyme that is used here, it is succinate, the same succinate, but we just put what? Dehydrogenase. Okay, so let me just use a different color here. So succinate, then dehydrogenase, like that. Okay, that is basically the enzyme, but we're going to produce what? FADH2. FADH2 from FAD. Okay, like that. So now here again, since we have produced the fumarate from succinate, again fumarate will undergo further processes to do what? To produce a different intermediate here, which is malate. Okay, so now fumarate will again produce what? Malate here. So this is what we call malate. Okay, so now this malate again, it will also undergo further processes to do what? To produce this intermediate. But before we talk about this intermediate, let's first talk about what? The enzyme that is used to catalyze this reaction from fumarate to malate. Okay, that enzyme again, it is very simple to remember because we are moving from fumarate to malate. So the enzyme is fumarase. Okay, the enzyme here is fumarase. Fumarase. That is basically the enzyme that is used. Okay, fumarase. And what are we producing here? So here we're not producing anything. Okay, but here we produce what? FADH2. Okay, from succinate dehydrogenase. Then here, since we have produced the what? The malate. Since the malate here has been produced, and it is 1, 2, 3, 4, 4 carbon atom molecule, and I said that from succinate up to this molecule, all the intermediates that are here, they're going to be having what? Same number of carbon atoms. Okay, so since this one is a malate, to move from malate to this intermediate, the enzyme that we're going to use is malate dehydrogenase. Okay, the same word malate dehydrogenase. Okay, dehydrogenase. Okay, then what we are producing here is what? We are producing this other intermediate, which is what? Oxaloacetate. Okay, oxaloacetate. This is oxaloacetate. Oxaloacetate. So now, this same oxaloacetate, for it to be made, this enzyme, which is malate dehydrogenase, since it is a dehydrogenase, here we are going to produce what? NADH from NAD+. Okay, so even here we have produced the what? The NADH. So NADH, okay, I can just write it here. NADH has been produced from what? Of course, it is happening here. Okay, it is happening here from NAD+. Okay, NAD+, like that. So let me just show that we are producing NADH from NAD+, where here malate to oxaloacetate, by which enzyme? Malate dehydrogenase. Okay, so now since we have produced the oxaloacetate, by the way, this process here, it is even reversible. Okay, it is reversible. Unlike this process here, which is having the alpha ketoglutarate, it is a reversible process. But this one is reversible. So now we have produced the oxaloacetate here. So this intermediate here that we have, since this is a cycle, okay, so this process now can now continue. Okay, it can now continue to do what? To do its work. So oxaloacetate will now combine with the acetyl CoA. Okay, no wonder I didn't complete like this this arrow here. This arrow here, it is showing that the oxaloacetate, this intermediate that has been produced from malate, can be converted to what? To citrate by combining with what? With acetyl CoA. So the moment acetyl CoA enters here, it will now be combined by what? Or with the oxaloacetate. Okay, like that, to be combined like that and produce the citrate synthase. Okay, and produce what? Citrate rather. Okay, by the help of citrate synthase enzyme. So this is now what will be happening. Okay, as we have seen now, the reason why they call it a Krebs cycle, because the intermediate that we're talking about here, it is the oxaloacetate. Okay, we start with oxaloacetate, it combines with the acetyl CoA, Then the process continues. Again, we produce oxaloacetate. Again, it combines with acetyl-CoA just like that. Okay. No wonder it is called what? A Krebs cycle. No wonder it is a cycle. But before I even end this lesson, we have to talk about the limiting enzyme. Okay. The limiting enzymes of this what? Of this Krebs cycle. Very important. You have to know it. The limiting enzyme here we have is the citrate synthase. So this is the number one. So we tick it. This is the what? The limiting enzyme that we have where in this process of Krebs cycle. Then we also have another limiting enzyme, which is what? The isocitrate. That is also another limiting enzyme. Then we also have, rather, the enzyme is isocitrate dehydrogenase. So this is not an enzyme, but it is a what? Intermediate. But this one, the isocitrate dehydrogenase, it is also the what? The intermediate. There are actually three very important intermediate, which are what? Limiting enzyme. We also have the alpha ketoglutarate what? Dehydrogenase. Okay, where did I write that? So here, the alpha ketoglutarate dehydrogenase. This one is also the what? The limiting enzyme in this process of what? Krebs cycle or TCA cycle. Okay, so these are the three enzymes that you have to take note as what? Limiting enzyme, because most of the questions that they ask examiners are going to ask maybe what are the limiting enzymes for this same reaction. You have to know them. Okay.